these have been changed. Both of these positions have been changed due to the methylation. And now, obviously, the donor, it no longer has the hydrogen, and it is now an acceptor. And this acceptor up here is actually now nothing with respect to hydrogen bonding. So we've obviously changed the acceptor-donor profile of this base. So this is nothing. So now O6-methyl-G, rather than base pairing favorably with C, it will also base pair not only with C, but with T. And this is what's shown here in this slide. The methylation position is neither a hydrogen bond donor or acceptor, and now we have the possibility of H bonding to either C or a T as demonstrated. So this is our C, and this is our T base. Okay, and these are, you know, both different configurations. Of H bonding patterns here. And we can see these different configurations because I've actually included over here the normal Watson-Crick GC base pair configuration as a comparison point. So again, because we've changed the base pairing capacity of this, you know, normally there is discrimination for correct GC base pairs, and now that discrimination is lost such that the G can pair with C some of the time, but you can also have the G pair with T some of the time, and that's perfectly acceptable. And now you can sort of appreciate how this alteration of base pairing through this damage event could then subsequently lead to mutagenesis. So basically, you know, our GC base pair can go through multiple rounds of replication so that we end up with an AT base pair. Okay, so just to reiterate, you can take a GC base pair through some steps as normal, and then finally end up with an AT base pair. Let's pause here and just summarize the first part of this lecture. So basically, I've distinguished between two classes of DNA damage. We've talked about loss of bases, alterations to bases, and oxidative damage to bases that can all occur spontaneously. We've looked at CBDs and alkylation that can be caused by environmental agents. It's also worth pointing out that alkylation damage can also happen from endogenous sources, and as I mentioned, there's a large pool of S-adenosyl methionine, so those reactions can have a propensity to go wrong and result in DNA base alkylations. So let me just write here, also endogenous sources, and again S-adenosyl methionine, or SAM as it's commonly known. So although alkylation is commonly environmental, it can be from endogenous sources. And the key thing with these various base alterations is that they change the base pairing properties of the bases, and thereby the fidelity of the information that is encoded in them. Alright, so we have a bunch of damage, but we don't really know what the context is, right? You know, if these various damages that I've shown you happen about 0.00001 time on average per cell life, it would be hard to care about them, right? But if they happened a billion times every second, then maybe you'd care about them too much. I don't know. So at any rate, we can play a guessing game. And the first thing I'll ask, you know, so alkylating damage, and in this figure I've shown S-adenosyl methionine alkylating a base. So this is SAM here, and our methyl group that's being added to adenosine. So just as a guessing point, how many alkylation lesions per average human cell per day would you think are occurring? Well, it is approximately 5,000 per cell per day. So in simple terms, it's actually fairly common. What about spontaneous depurination? So pretty similar order of magnitude at about 10,000 events per cell per day. Interestingly enough, depyrimidination is only about 500 per day. So depyrimidination is on order of magnitude about 500 per cell per day. And that just has to do with the differences in the inherent stability of the glycosidic bond in both cases. So difference is due to stability of glycosidic bond. Deamination, if you just consider cytosine to uracil, is about 100 bases per cell per day. So deamination, so if we just consider C to U, is about 100 bases per cell per day. So all of these damages occur even in the absence of an external perturbation at a very high frequency during the average day of an average cell. And so certainly if you're having this kind of damage occurring at such a high frequency, you have to have some means to address this type of damage. And let's not forget about Mr. Golden Sun. Mr. Golden Sun says, I'm going to fry all your pyrimidines. So just put a sad face on him right here. So, you know, at your skin during peak hours of sunlight, you approximately have about 100,000 lesions occurring per cell per day. Ouch, you know, that is a lot. So really, if you think about it, nature is not our friend. In order to prevent nature from killing us, then, we have different DNA repair systems to address these different forms of damage. There are a lot of different repair systems, but I'm only going to talk to you about some of them. And today we'll talk about direct repair in this lecture. I'll also talk about base excision repair, and I'll talk about nucleotide excision repair. We are not going to talk about mismatch repair or post-replication repair due to time constraints. But if they are of interest to you, let me know and I can suggest some independent reading. And then in our upcoming lecture, we'll talk about double-strand break repair. So that's the next lecture. All right, so the repair systems we are going to go through, they're going to be in sort of order of complexity. And the most simple repair system is referred to as direct repair. And in direct repair, the damaged base is repaired in the DNA in a single step. So it is essentially just a reversal of the DNA damage. Okay, so just a reversal of the DNA damage. And as we'll see in a moment, other repair systems often involve the removal of the damaged base and then resynthesis to allow insertion of an undamaged correct base. The direct repair system does not remove the damaged base. It actually just fixes it in the context of the DNA. So, just fixes it in context of the DNA. Okay, and so on this slide, again, I've depicted O6 methyl G. So here's our methyl group. And as a fairly common alkylation damage, it has a dedicated repair system, and that is catalyzed by an enzyme called MGMT, all right, and a one-step suicide reaction. So maybe you can write suicide here, so a suicide reaction. And the idea is simple in that you have this methyl group that, I've depicted up here that you want to get rid of. The MGMT has an active site cysteine, which is right here, so it's labeled in blue. And the cysteine simply just catalyzes the transfer of the methyl group, so this, onto that cysteine residue. So it goes from the guanine to the enzyme. And then once this happens, the enzyme is essentially inactive. Okay, so the methyl group transfer inactivates the enzyme. Okay, so is this a costly scenario? Yes, it is, because it means each time you synthesize this protein, it can repair exactly only one alkylated base, and then the protein has to be degraded and its components recycled. It also has this sort of interesting kind of knock-on effect, where if you consider a cell and what a cell's capacity might be to repair O6-methyl G, its capacity relates directly to how much MGMT it can make. So cells that express a high level of MGMT are quite resistant to this alkylation damage, whereas cells that express a low level of MGMT are quite sensitive to alkylation damage. And of course, you might imagine that then you could have consequences if, for example, you're considering treating someone, say a cancer patient, with an alkylating agent. You might want to consider what their normal cellular capacity might be to repair that kind of DNA damage. The MGMT reaction is a very simple one, and it's shown here. This is the active site cysteine in MGMT, so the active site. And a little nucleophilic attack thing on this methyl group. So cysteine nucleophilically attacks the methyl group, resulting in the transfer of that methyl to the active site cysteine, which then essentially inactivates MGMT. So inactivates MGMT, and then restores the normal G back to its original form. Below is actually a structure of MGMT, which is actually shown in blue here. Okay, so this is MGMT, and alongside a piece of DNA with a single O6-methyl G residue. So here's our DNA with the O6-methyl G residue. This structure was done by John Tainer. So John Tainer, and when he was at Scripps, and now he's at MD Anderson. The enzyme is pretty cool, and the way it works, I think, is also pretty interesting. So let me just spend a couple of minutes orienting you on this. So again, we have our DNA, right? MGMT is over here. And what I want to draw your attention to is this sort of little green bit of protein. And I'll show you a different view in a minute that I think might actually make it a little bit clearer. But there's an arginine residue here that is actually poking into the DNA double helix from the minor groove side. And the result of this amino acid sticking into the helix is that the damaged base, which is actually seen here, is actually rotated from its normal base paired position within the helix, such that now it's extra helical. Okay, so there's an arginine that pokes into the helix from the minor groove. And what this does, the base flips outwards into the active site of MGMT. Okay, so technically, the base should be sitting in here with all its friends, but now it's kind of like out here. And where is out here is the active site of the MGMT enzyme. So the enzyme basically kind of pokes a finger into the helix, rotates the damaged base out into its active site, where it can then catalyze through nucleophilic attack this methyl transfer reaction. So a pretty cool arrangement. Here's a couple of other views of the same thing. So this is just kind of this view here, a side view, and you can sort of see this, interdigitation and the base actually flipping out. Okay, so here is that that arginine residue right in here that's poking into the helix. Okay, so it's basically flipping the base extra helically, and again, if we view it from the top down of the helix, it's pretty wild because you can actually see that the arginine is intimately associated with within the center of the helix, right? So it's kind of stuck right in there. So in addition to causing the damaged base to flip out, the positioning of the arginine is such that it's kind of stuck in the middle of the helix now, and also forms hydrogen bonds with the C residue that the damaged G used to be hydrogen bonded to. So not only does it flip the base out, this arginine residue, it can then be used to form hydrogen bonds with the orphan base, such that it can sort of stabilize the normal double helical structure, even though one of its bases has been flipped out into the active site of the enzyme. So a pretty cool arrangement for repairing damage in situ. And it turns out, although this is not always the case, there are a large number of DNA repair enzymes that use this kind of base flipping strategy, either to identify damage or to repair damage. The second system that I want to talk to you about is called base excision repair. And the idea with base excision repair, or BER, is that we're going to identify a damaged base, we're going to remove just the base and leave an AP site. And then we're going to process the AP site to remove the sugar that is left at the abasic site. And this results in a 1 nucleotide gap. And then we're going to use a polymerase to fill it in so that one missing nucleotide is replaced, and then a ligase will be used to seal it. So we're going to remove the damage. We're going to use the sister strand as a template to reinsert the correct base. So this basically contrasts with the previous system where the damage is repaired and reversed in situ. Here, we're actually removing the damaged base and replacing it entirely. So I'll first take you through a schematic, and then we'll talk about these steps in a little bit more detail. The whole pathway is depicted on this slide. The base excision repair is actually limited by the presence of specific DNA glycosylases. Okay, so this is limited by a specific DNA glycosylases. Okay. And what do the DNA glycosylases do is that they recognize the damaged bases and they cleave the N-glycosidic bond between the base and the sugar. Okay, so they recognize the damaged bases and cleave cleave the N-glycosidic bond between the sugar and the base. So this cleavage reaction is exactly the same as the spontaneous reaction that occurs in the formation of an AP site that we discussed previously. It's just that this one is actually enzyme catalyzed. So you might guess from an enzyme catalyzed versus a spontaneous reaction, this is going to have a way higher reaction rate. The downside, and the reason I say limited, is that each glycosylase basically recognizes one kind of damaged base. Okay, so each glycosylase, I'll just write this over here. So each of them only recognizes one damaged base. And so this means you can fix a base damage as long as you have a specific glycosylase for it. So if you have some new alien form of base damage that may arise, there would be no system present to recognize it because the whole pathway depends on the action of these specific glycosylases that we're going to talk about. And plus, if you have different kinds of base damage that arise at high frequency, you can devote these specific enzymes to repair those specific damages, but it makes the system limited in terms of the scope of what kinds of damage it can deal with. So it's sort of like the direct repair system, and is in fact, it's even more limited, right? Because literally one protein is responsible for repairing one kind of damage. This pathway encompasses more forms of damage, but it comprises multiple different kinds of glycosylases. Okay, so the first thing in this schematic, if we look at point number 1, which is depicted here, is the glycosylase recognizes the damaged base and cleaves the glycosidic bond to release the damaged base. This results in the formation of an AP site. So we already talked about this, and here's the AP site, okay, which we already know is bad, right? So this is kind of a curious thing. We've gone from base damage, which could cause mispairing, to an AP site, which has no information, blocks polymerization, and can result in backbone cleavage. So this kind of seems like a serious step backwards, and I think, you know, a general lesson that one might take home from this is that once you initiate this repair pathway, you should be strongly motivated to finish it, right? Because the intermediates in the repair pathway are themselves quite mutagenic. Like obviously an AP site is mutagenic. And this kind of gets worse because after we have our AP site, the first thing we're actually going to do is make a backbone cleavage. So it's like, oh my goodness, we have an AP site and then we cut the backbone. Are we insane? So the backbone is cleaved by an enzyme called AP for apurinic or apyrimidinic endonuclease. Okay, so this is step number 2. And it cleaves on the 5' side of the AP site. Okay, so if we're looking at our strand, remember this is 5' over here and this is 3' over here. And again, it's called an AP, 5' AP endonuclease. Okay, so now you have a backbone cleavage. You've got a nick, and this generates a 3' hydroxyl, right? So here's our 3' hydroxyl. Okay. The problem now is that adjacent to the 3' hydroxyl, we have a 5' right here, a 5' deoxyribose phosphate. 5' deoxyribose phosphate. Phosphate. So, you know, we don't have an actual gap. We've got a nick, a sugar, and then the rest of the chain. So we need some further processing before we're in a position where a polymerase can insert the correct nucleotide. So what happens at this stage, at stage number 3, is that we need an enzyme that will remove the 5' deoxyribose phosphate. And this is performed by an enzyme with a 5' DRP lyase activity. Okay, so it's represented here. And so we have a 5' phosphate, and once this cleavage happens, we're left with that 3' hydroxyl. We've now got a 5' phosphate, and we've released the 5' deoxyribose phosphate. So this is the 5' deoxyribose phosphate. So now basically we're left with a 1 nucleotide gap. So this is perfect, right? We have a 3' hydroxyl, and again, remember, a polymerase can use this. We've got a nucleotide gap, so a 1 nucleotide gap, and a 5' phosphate, which we depicted over here. And keep in mind that although I'm showing this all in the context of a double strand, this is all in the context of double-stranded DNA, right? So all in the context of double-stranded DNA. So this 1 nucleotide gap is then annealed to the template, and that encodes the correct information for resynthesis. The same as the spontaneous reaction that occurs in the formation of an AP site that we discussed previously. It's just that this one is actually enzyme catalyzed. So you might guess from an enzyme catalyzed versus a spontaneous reaction, this is going to have a way higher reaction rate. The downside, and the reason I say limited, is that each glycosylase basically recognizes one kind of damaged base. Okay, so each glycosylase, I'll just write this over here. So each of them only recognizes one damaged base. And so this means you can fix a base damage as long as you have a specific glycosylase for it. So if you have some new alien form of base damage that may arise, there would be no system present to recognize it, because the whole pathway depends on the action of these specific glycosylases that we're going to talk about. And Plus, if you have different kinds of base damage that arise at high frequency, you can devote these specific enzymes to repair of those specific damages, but it makes the system limited in terms of the scope of what kinds of damage it can deal with. So it's sort of like the direct repair system, and is in fact, it's even more limited, right? Because literally one protein is responsible for repairing one kind of damage. This pathway encompasses more forms of damage, but it comprises multiple different kinds of glycosylases. Okay, so the first thing in this schematic, if we look at point number 1, which is depicted here, is the glycosylase recognizes the damaged base and cleaves the glycosidic bond to release the damaged base. This results in the formation of an AP site. So we already talked about this, and here's the AP site, okay, which we already know is bad, right? So this is kind of a curious thing. We've gone from base damage, which could cause mispairing, to an AP site, which has no information, blocks polymerization, and can result in backbone cleavage. So this kind of seems like a serious step backwards, and I think, you know, a general lesson that one might take home from this is that once you initiate this repair pathway, you should be strongly motivated to finish it, right? Because the intermediates in the repair pathway are themselves quite mutagenic. Like obviously an AP site is mutagenic. And this kind of gets worse because after we have our AP site, the first thing we're actually going to do is make a backbone cleavage. So it's like, oh my goodness, we have an AP site and then we cut the backbone. Are we insane? So the backbone is cleaved by an enzyme called AP for apurinic or apyrimidinic endonuclease. Okay, so this is step number 2. And it cleaves on the 5' side of the AP site. Okay, so if we're looking at our strand, remember this is 5' over here and this is 3' over here. And again, it's called an AP, 5' AP endonuclease. Okay, so now you have a backbone cleavage. You've got a nick, and this generates a 3' hydroxyl, right? So here's our 3' hydroxyl. Okay. The problem now is that adjacent to the 3' hydroxyl, we have a 5' right here, a 5' deoxyribose phosphate, 5' deoxyribose phosphate, phosphate. So, you know, we don't have an actual gap. We've got a nick, a sugar, and then the rest of the chain. So we need some further processing before we're in a position where a polymerase can insert the correct nucleotide. So what happens at this stage, at stage number 3, is that we need an enzyme that will remove the 5' deoxyribose phosphate. And this is performed by an enzyme with a 5' DRP lyase activity. Okay, so it's represented here. And so we have a 5' phosphate, and once this cleavage happens, we're left with that 3' hydroxyl. We've now got a 5' phosphate, and we've released the 5' deoxyribose phosphate. So this is the 5' deoxyribose phosphate. So now basically we're left with a 1 nucleotide gap. So this is perfect, right? We have a 3' hydroxyl, and again, remember a polymerase can use this. we've got a nucleotide gap. So a 1 nucleotide gap and a 5' phosphate, which we depicted over here. And keep in mind that although I'm showing this all in the context of a double strand, this is all in the context of double-stranded DNA, right? So all in the context of double-stranded DNA. So this 1 nucleotide gap is then annealed to the template and that encodes the correct information for resynthesis of that nucleotide. So then in step number 4, all we need is the polymerase, because we basically, if we were to draw this as a template, we have a primer, we've got our 3' hydroxyl, we got our 5' phosphate, and we got our 1 nucleotide gap. So all we need to do really is just insert that 1 nucleotide. And we know that the prime template is a good substrate for DNA polymerase. So once we reduce the damage to that position where we have a 3' hydroxyl, a 5' phosphate, a single nucleotide gap, DNA polymerase simply has to catalyze the insertion of 1 nucleotide, and that leaves a nick, and then DNA ligase can come in and seal that. So we have the ligase that comes in, and that's all in step number 4. So this slide is just showing a little bit more detail of the glycosylase step. So again, we'll just talk about more in depth about the glycosylase step. And again, it's just a simple cleavage of the N-glycosidic bond between the deoxyribose and the damaged base. And of course, the damaged base gets released. So, second thing in this figure, we're going to have cleavage of the damaged base. And this again is a hydrolysis reaction. And we release that damaged base. Now, in this context, what happens next is the AP nuclease. So step number 4, the AP endonuclease cleaves 5' to the damaged base, right? So we're going to get that 5' cleavage, and that would leave you with a 3' hydroxyl as promised, right? So this becomes a 3' hydroxyl, and the 5' end would get cleavage by the lyase. Okay, so again, we're going to have step number 5, in which the lyase comes in, and this will subsequently release the 5' deoxyribose phosphate, and that's what gives us the context where we have a 1 nucleotide gap flanked by a 3' hydroxyl and a 5' phosphate. So what else do I want to say here? So this example is one of which we're getting rid of uracil and DNA. Okay, so the example is getting rid of uracil from DNA. And so a lot of what base excision repair does is remove uracil from DNA. And part of the reason that it does so is that there's lots of spontaneous deamination of cytosine. So lots of spontaneous deamination of cytosine, which of course would then be highly mutagenic if the deaminated cytosine was allowed to remain as uracil in DNA. So cells very actively remove uracil from DNA, and a thing I guess I already emphasized this is that this hydrolysis of the glycosidic bond is again the same as the spontaneous reaction. It's just that it's enzyme catalyzed, and so it occurs at a much higher rate. And for this reaction here, the enzyme responsible is uracil DNA glycosylase. As I mentioned, the whole process of BAR is limited by the presence of specific glycosylases. And so I've listed in this table a number of human glycosylases, and I want to show you them as a theme so that you can appreciate what kind of damage this pathway is used to address. So the ones that are at the top, all in blue, are these ones here, and they're specialized for removing U. So they remove U. So a lot of the pathway's functionality, as you can see, is devoted to removing uracil and DNA. This one down here is devoted to removing different methylated damages. So methylated damage. And again, it's really small alkylations that these ones are responsible for. They're just very simple, they're not complex alkylations, okay? And then at the bottom here, there's a couple devoted to removing 8-oxo-G. Okay, so these ones remove 8-oxo-G. And so the pathway removes some very important forms of DNA damage, but it's by no means a universal pathway. It tends to be restricted to two small modifications of bases, so things like deamination, methylation, oxidation. Okay, so key thing here is restricted to small modifications. Of bases. We'll finish BER off by taking you through the last bit of the pathway. So we've talked about the glycosylase removing the base, cleavage 5' by AP endonuclease, and then of course I noted at the point where we have this 5' deoxyribose phosphate that we need to get rid of, and the 5' DRP removal requires a lyase. Okay, so the key thing is that 5' DRP removal requires a lyase. Oops, a lyase. And in eukaryotic cells, the lyase functionality is actually provided as part of a DNA polymerase called DNA polymerase beta. And it's depicted here. Okay, so poly beta is a specialized polymerase. And not only does it have the normal 5' to 3' DNA polymerization capacity, it also contains the 5' deoxyribose phosphate lyase activity. Okay, so it also contains the 5' DRP lyase activity. All right, Alright, so in the typical mode of basic excision repair, which is the one I'm going to describe, which is called the short path basic excision repair, after the initial incision by AP endonuclease, the lyase removal of the 5' deoxyribose phosphate is done by DNA polymerase beta. Polymerase beta, since it's a polymerase, is then in position to catalyze the synthesis of that one nucleotide polymerization to fill in the gap. So we have a gap filling here, and again, this occurs right here. So that's done by the polymerase, and then what remains after gap filling is a nick. And the nick is then readily sealed by a DNA ligase. So the nick is sealed by the ligase. So this is actually termed short patch, or SP, because only one nucleotide is replaced. So only one nucleotide is replaced. There does exist over here in this this fuzzy region a second pathway that is called long patch, whereby a greater stretch of nucleotides is resynthesized, but the predominant pathway that's used in mammals is the short patch pathway. Okay, so this is the predominant pathway in mammals. Okay, so that summarizes basic excision repair. Alright, so the next and final repair pathway I'm going to talk about is nucleotide excision repair, or another terminology or acronym for it is NER. So this kind of takes the complexity up a bit more, sort of an additional level, because nucleotide excision repair is a more general mechanism for repair. In contrast to the ones we've seen previously, NER does not recognize damaged bases. Okay, so it doesn't recognize damaged bases. In contrast, what it does recognize is either distortions of the DNA helix or disruptions of hydrogen bond. Okay, so it's a more general mechanism for repair. And it either recognizes distortions of the helix or disruptions in hydrogen bonding. Okay. So rather than looking for specific changes in the bases themselves, what this pathway looks for is sort of the downstream consequences of those changes. So as I noted, like for example in the case of UV damage, there's a great disruption of the helical structure of DNA and disruption of the hydrogen bonding because of this new sort of spatial constraints that are imposed, for example, by a CPD or a 6,4 photo product. So nucleotide excision repair would not be recognizing a CPD directly, it would be recognizing the effects of the CPD on the structure and stability of the DNA helix. So this pathway is more general than the two that we've previously described, and it's extremely important for UV and damage and bulky adducts. Okay, so it's important for UV damage and bulky adducts. And what I mean by bulky adducts could be products of more complex alkylation damage, for example. And what is sort of nice about this is that you then have a system that can respond perhaps to chemical forms of damage that it hasn't seen before, as long as those chemical forms of damage can cause a disruption of hydrogen bonding or distort the form of the double-stranded helix. Okay, so this is a very broad general schematic of the steps, and the illustration really is with a pyrimidine dimer that's depicted here, probably caused by UV damage. And the way NER works is that the damage is first identified. There's a specific incision of only the damaged strand in two positions flanking the DNA damage. Okay, so that's in step number 1. We get incision 1 over here and incision 2, and it encompasses that specific damage. This damaged oligonucleotide is then removed, and that occurs in position 2. So we've basically taken that damaged area and cut it out. And then in step number 3, which is depicted here, the DNA polymerase reaction occurs, and the DNA polymerase fills in the gap. So again, the template for this damage, or the template, for the DNA polymerase reaction is the strand to which the damaged strand was originally annealed to. And so you're using the correct template to resynthesize the damaged area. Okay, so again, you'll use the correct template to resynthesize the damaged strand. So let's look at NER at a more granular level, and we're going to focus on NER in humans. So this is where things actually get a bit complicated. So we can start with a lesion. So it's depicted here. And this lesion causes or disrupts base pairing, or even distorts the helix in some way. This lesion is then bound by dimer proteins that recognizes the damage, and the damage recognition dimer is composed of XPC and HR23B, which is this dimer here. So it's a dimer that recognizes the damage. Okay, so they're both for damage recognition and binding. So when the damage is recognized, the dimer causes an increased kinking of the DNA. Okay, so let me just write this over here, XPC and HR23B. When they recognize that damage, it actually kinks the DNA. And this is important because the DNA bending or kinking is actually a signal for the recruitment of a complex called TF2H. Okay, so this is a signal for recruitment of TF2H. TF2H is a multi-subunit complex. And why is this important? Because it's actually composed of two DNA helicases. And these two DNA helicases, so two DNA helicases are XPB and XPD. And they're represented here in the schematic. And what do DNA helicases do? They pull the strands of the DNA apart, so they unwind the DNA. So after recruitment of these DNA helicases, there's a helix unwinding step. Okay, so these two will then unwind the DNA. And so what happens at the helix unwinding stage is that the helicases will unwind until one or the other hits the lesion. So they unwind the DNA until one or the other helicase hits the lesion. So really, this unwinding kind of serves as sort of the lesion verification step. Once one helicase hits the lesion, the second helicase will continue to unwind until of about 20 base pairs are unwound. So one hits the lesion, the second one will keep unwinding until approximately 20 base pairs are unwound. And that's depicted in figure C of the schematic. So now we have a scenario where we have our lesion and it's kind of extruded as a single-stranded DNA loop and is now dissociated from the non-lesion containing strand. So our single-stranded DNA loop. All right, continuing on with NER. So what would you imagine would happen to the single-stranded DNA that doesn't contain the lesion? So I'm just going to highlight it here in this slide. What does it say here? It says RPA. So that, of course, the RPA is readily bound and bound to that strand that doesn't contain the lesion. And again, because remember, cells hate single-stranded DNA. They don't like it to be around. So once that unwinding has occurred, which this catalyzes in addition to the protection of the undamaged strand, because we already have the damage, we don't want to make more. So it's coded by RPA. And then this follows the engagement of this complex that's called XPG plus XPA. And what this is a nuclease. Okay, so let's just reiterate this. The single-stranded undamaged strand is bound by RPA. And this leads to the engagement of XPG plus XPA. And again, as I stated, this is a nuclease. Following this, the engagement of XPA and XPG, we then have the promotion of the entry of XPF, which is also a nuclease. So then this promotes recruitment of XPF, which is also a nuclease. And so we have this whole thing assembled here. And once it's all loaded, we can have a double incision made that cuts the damaged strand out. So one nuclease cuts on one side of the lesion, the other nuclease cuts on the other side. So now the damaged base can be removed, and I guess for completeness sake, we show here that XPF cleaves 5' of the lesion. So this cleaves 5' of lesion, And as shown on this side, XPG and XPA cleave 3' of the lesion. So, and then now what we're really essentially left with is a prime template, right? So we'll get that removed. And now we've got a 3' hydroxyl. We then got some more double-stranded downstream. We got our template. So this is a perfect, substrate for DNA, for the DNA polymerization reaction. And in fact, because this is more than a single nucleotide gap, this actually gets repaired by the entire DNA replication machinery. Okay, so it's a substrate for the polymerization reaction, and it gets repaired by the DNA replication machinery. And as shown, the primer terminus is a fantastic substrate for binding of, the clamp loader, RFC, which then catalyzes the loading of pCNA, which then serves as a processivity factor for poly delta, which is shown here, and then poly delta fills in the gap, leaving a nick in which the ligase can then cause sealing of that gap, of that nick. And finally, to conclude NER, what I want to do next is actually just show you a bit of the structural stuff. The issue really is, how do we recognize the DNA damage without physically recognizing the damage itself, as in the damaged base? So how might we go about to do this? And the way it works was actually suggested by this crystal structure, which is from Nick Pavlicek's lab at Sloan Kettering, and it's a structure XPC and HR23B. And so those are the damage recognition and binding protein complexes that are at the top of the schematic of the nucleotide excision repair pathway. And in this figure, the complex interacts with DNA containing a CPD. Okay, so basically we have a crystal structure of XPC plus HR23B. And that's all this colored stuff here. Okay, so all of this here is XPC plus HR23B. And then the gray is the DNA, which I'll label over here. And then we can see our CPD down in this region. And there's sort of two things interesting about this structure. And one is, hopefully you can see this little red loop, and I'll draw a circle around it. It's stuck straight into the helix. So it has the same sort of theme of poking a protein finger into the double helix. And the result of this is that there has been base flipping that happens. And these are demonstrated by these darker colored bases. So you can see them here. They're flipped out of the helix. So essentially it's almost as inserting a protein finger into the helix, flipping out a couple of bases, and that all sounds good for a pyrimidine dimer. Okay, so this is stuck. This region is stuck in helix. And what it leads to is base flipping. And what's curious about this is that in contrast to the other example we saw, it's not flipping the damaged base into an active site, because this is just a recognition enzyme. Okay, so the base flipping is not into an active site. Okay, and again, it's not physically catalyzing repair. It just actually flips the damage away from where the enzyme is. So the enzyme is not doing this base flipping as a mode of binding to the damage. It's actually doing the base flipping as a means of probing for the stability of the DNA helix. Okay, so it's probing stability of the DNA helix. Okay, and so the idea with XPC and HR23B is that this beta as a hairpin turn piece of protein basically probes the integrity of the DNA. Okay, so it's probing the stability or the integrity. So it's like walking along the DNA. Can I insert the protein complex into the DNA? If I can I then flip out the bases? And this base flipping is then what actually leads to the conformational change that adds that additional kink to the DNA that then promotes all the downstream protein recruitments. So this then leads to the additional conformational change, which then leads to the recruitment of other proteins. Okay, so basically only if base pairing is disrupted is XPC able to flip out the bases and then kink the DNA. And this kinking of the DNA is then a prerequisite for all the other steps. Okay, so again, it's just for recognition. And only if base pairing is disrupted is XPC and HR23B able to flip out bases and kink the DNA. Okay, again, XPC is essentially acting as a helical stability probe. So it's another way of describing it as a stability probe. And so again, it wanders along the DNA, asks is the hydrogen bonding normal, is the stability normal? If the stability is weaker than it should be, then XPC is able to insert itself into the DNA and bend the DNA, and then this catalyzes the downstream recruitment events. So that's the way the system is able to detect DNA damage without detecting the damage itself. It detects the downstream effect of the damage on the helical stability of the DNA. Pretty amazing. Let's just summarize now everything that we've covered thus far, and we did go into detail about how direct repair reverses specific lesions in a single step. We talked about BER and how they use, in this pathway, specific glycosylases to initiate repair of specific base modifications. And finally, we ended off with NER, which detects DNA helix destabilization caused by diverse lesions. And this concludes the DNA repair lecture.